Our next speaker will be Mario Benn, who will present on an exciting microtissue platform to study the reciprocal crosstalk between cells and their extracellular matrix. The floor is yours, Mario.
Thank you very much, Viola. So welcome everybody here in the hall and also in the cyberspace online in the stream. Many thanks to Beat and Viola and the organizers to inviting me, having the chance to share my work. It's my pleasure to present our de novo microtissue platform today to you. I personally couldn't agree more with the spirit of this summit. We are facing great challenges in clinics, and interdisciplinarity is key to solve them. I personally gained plenty of clinical experience in human and veterinary medicine, and the latter I focused on surgical oncology. I conducted in vivo research in large animal models, namely in sheep, goat, mini pig, and others. But soon my curiosity led me to investigate mechanobiology and to become a tissue engineer. As a surgeon, I mainly dealt with things that I can touch and see. As a tissue engineer, I deal with much smaller length scale. And this shift in perspective is absolutely striking and allows us to bridge many more different gaps. So I'm fully convinced both interdisciplinarity and bridging length scales is key to translate our knowledge towards clinics. And luckily, what we see at Clinam, there's a huge community working with this aim worldwide. In my tissue engineered system, I investigate how to control myofibroblasts in their dynamic ECM environment. But what are myofibroblasts? These are mesenchymal cells. They produce lots of fibrous ECM and apply high contractile forces to their environment, so you can see them like little Arnold Schwarzeneggers in the tissues. With this, they are key effector cells in wound healing, but can also lead to imbalanced ECM remodeling in fibrosis or cancer. So they can be both friend or foe. In healthy wound healing condition, they help to close the wound and disappear when they are no longer needed. Under disease condition, however, they remain on site, which can then lead to further myofibroblast activation, resulting in a vicious circle of fibrosis with vital organ failure. The myofibroblasts differentiate up on different triggers, for example, mechanical tension or TGF-beta, and from 2D cell culture, TGF-beta is known as the prominent activator of myofibroblast. However, targeting TGF-beta in clinical trials failed so far miserably. This underlines that this activation of fibroblasts is tightly bound to the three-dimensional environment, and we aim to identify what factors lead to stabilization or clearance of myofibroblast. We are extremely happy that our recent paper was featured as cover in Science Advances this year. In a nutshell, in our platform, we seed dermal fibroblasts and grow de novo tissues. So we seed fibroblasts, but the tissues mainly show myofibroblasts in their young growth front, which you can see by the orange marker in the growth front on this cover image. The growth front is highly stretched, proliferative, and dominated by fibronectin protein. Over time, the tissue matures, and the myofibroblast revert back to fibroblast, which can be found in the mature core tissue dominated by collagen. So the spatial mapping of stained microtissues allows us to retrieve temporal information of the dynamic cell ECM interactions, like, for example, the myofibroblast to fibroblast transition. And I'm very happy to walk you through the details in the following slides. Our approach is based on PDMS scaffolds with millimeter-sized clefts. We fabricate the substrates by replica molding of negative master structures, and PDMS scaffolds are then covalently functionalized with fibronectin to support the cell adhesion. Primary human dermal fibroblasts are then seeded on top of these substrates, and they first form a confluent multi-layer of cells on the horizontal plane and the vertical plane of these scaffolds. Then they start to grow and advance into the third dimension, and the third three-dimensional tissue is formed into this open cleft. About 2 weeks later, they have then formed a complete tissue that is spanned in between the clefts, similar like a spider web in the corner of a room. We found biomarkers indicating the outer shell of the microtissues, which you find here in these cross-sectional slides depicting fibronectin and tenascin C. Previous work demonstrated that these microtissues grow towards the center of the cleft in a layer-by-layer manner. For example, labeled fibronectin supplemented at day 4 and day 10 is locked within the growth front at the time, and after further tissue growth, the two differently labeled fibronectin, which you see here in yellow and here in red, can still nicely be separated. And that means we get a gradient of maturity, and we can translate the spatial to temporal information in one and the same microtissue. A very important fibroblast and a very important feature hereby is the fibroblast to myofibroblast transition, which occurs in the proliferation zone and happens without exogenous TGF-beta. We identify myofibroblasts by alpha-smooth muscle actin immunostaining, which can see to be highly expressed in this growth front. While the tissue matures, the myofibroblasts revert back to fibroblasts, so we can find the growth front dominated by myofibroblast and the core tissue dominated by fibroblast. Interestingly, the TGF-beta supplementation cannot convert the resident fibroblast in the tissue core to myofibroblast, which you can see here on the right. So supplementation of TGF-beta appears in a slightly widened alpha-smooth muscle actin growth front compared to the control, but does not revert the resident fibroblast to myofibroblast. And this suggests that a mechanical trigger can't be overwritten by chemical stimulation. Here we see the myofibroblast stained by alpha-smooth muscle actin in orange, accumulating in the growth front, which is also rich in fibronectin. We found that during the tissue maturation, the alpha-smooth muscle actin signal decays, while now mature collagen fibers detected by second harmonic generation increase. And in the profile plots here on the right bottom, you see that it is a profile plot along this horizontal middle line. There's a steep spatial gradient between the fibronectin-rich growth front and the collagen maturing tissue. We also identified tenascin C. Tenascin C is a matricellular protein found in sites of remodeling. And tenascin C is also decorating the fibronectin fibers in the growth front. Therefore, myofibroblasts are stabilized in regions rich in fibronectin and tenascin C. And we also identified an important transition zone in which all the growth front markers decay and the collagen levels take over. And this indicates that this transition zone is also crucial for the myofibroblast to fibroblast transition. We further tested our bacterial binding peptide, which has a higher affinity to relaxed fibronectin, which you can see on the top here. Once fibronectin gets stretched, peptide binding decreases, and this allows us to probe the fibronectin fiber stretch in tissues. On the right, in green, you see the total fibronectin levels stained, and in red, the peptide probe labeled with Cy5. And yellow is where then both signals co-localize. Overall, the growth front contains dominantly stretched fibronectin, and these are the regions which co-localize also with alpha smooth muscle actin in the growth front, and therefore myofibroblasts are stabilized in a growth front consisting of stretched fibronectin. We also tested different pharmaceutical inhibitors. In the matter of time, I cannot go in detail here, but let me summarize as follows. All the tested inhibitors caused either a delay or an incomplete conversion of myofibroblast to fibroblast and tissue maturation, which you see exemplarily depicted here on the right compared to the control on the left. So you can see both in the confocal images, but also in the profile plots, the growth front marker decay much more slowly compared to the control. Our research paints a vivid picture of a dynamic dance between the cells, depicted in orange on the left, and the ECM, depicted in blue on the right, and every move needs to be precisely timed. Taken together with the literature, we identified a series of interactions between the cells and the ECM, which are crucial for tissue growth. So these interactions can be spatially assigned to the growth front, the transition zone, and the core region. In the growth front, we find high cellular contractile forces stimulating profibrotic gene expression, including tenascin C. The fibronectin fibers are stretched by the cellular contractile forces, and the cell secretion of tenascin C and tissue transglutaminase leads to cross-linking, which stabilizes this early produced fibronectin ECM. In the transition zone, tenascin C inhibits non-cellular contractility, and highly stretched fibers transition to relaxed fibers, and relaxed fibronectin fibers template for collagen fiber assembly. As the tissue matures, the cells finally sit in a collagen-dominated ECM, and the cells lose all their alpha smooth muscle actin signal, and the myofibroblast to fibroblast transition is then completed. Let me summarize the strengths of the platform. Relevant cues for spontaneous tissue growth are integrated. It features varying gradients, enables phenotype transitions. There's no hydrogel interface needed to advance into the third dimensional space, and the tissue-to-tissue variation is minimal. It features live cell microscopy and long-term incubation, and it provides the potential to support 3R principles, which is very important. This means not only less animals suffer through replacing, refining, and reducing the animal experiments, but also avoiding the huge translational gap between the species. In the outlook positioning, we plan to map the spatial proteomics and transcriptomic signatures of the varying regions. We aim to alter tissue tension and translate the system towards clinics. For example, by integrating cells from different organs and also from disease states. Also by implementing compounds, for example, nanoparticles that could directly target specifically zones in the microtissues, and we would then be able to monitor real-time dynamic ECM cell interactions. So, as I said in the beginning, interdisciplinarity is key to tackle our life science challenges. And I always love to create new synergies by combining multiple methods. So if you got ideas how to collaborate, feel free to reach out. I'd like to finish with a quote from Isaac Newton. if I have seen further, it is by standing on the shoulder of giants. This describes wonderfully that we all rely on the work and on the help of others on the path to move forward. And therefore, I'd like to thank everybody who contributed to my research, especially Viola, my colleagues, my students, and of course, the respective funding. You for your attention, and I'm happy for taking your questions now or later.